One.
Hey everyone, it's Mrs. Shannon, and this is the Macromolecule Prezi Screencast for the second part of our notes going on here. So hopefully you guys are hanging in there and you have the PDQs in front of you. You've had a chance to look at them so you know what to look for. So the title of this Prezi, again attributed to David Nefke, is Macromolecules, also known as How to Make a Hairless Cat and Every Other Living Thing. And the reason, except the picture's not showing here on this one, I'm sorry. You can't see the hairless cat. I'll show you a picture in class. The reason it's how to make a hairless cat and every other little thing is because we're going to study macromolecules, which are sort of the building blocks of everything, especially one category, proteins. Hopefully you guys have remembered hearing about proteins and how cool they are and how they function. Oh, there we go. There's our warm, bald cat belly. And so we're going to see how these proteins are actually structured as well as other macromolecules. So big questions of this presentation. How are the molecules of biological systems constructed? So how are they made? What are the particular groups needed for different systems, and how do the interactions lead to the emergence of life functions? Again, that word emergence is going to be coming out. You're probably going to get sick of it by the end of the year, but it's how new properties come about from the building blocks, how they're different than the building blocks that put them together. So what's a macromolecule? I actually am not a big fan of the term macromolecule. They call it macro because it's bigger molecules, even though they're quite tiny. It's made of a few common atoms, and most of them are made out of the element CHNOPS. They are very complex. They're also known as biomolecules. You hear them called organic molecules. I've been referring to them in the last couple of years as biomolecules instead of macromolecules, but you'll see them both ways. Oftentimes when we're looking at these, we need models. They are, even though they're macro, they're still quite tiny, and the shape of them, to really look at the shape, we need to use different modeling software to see the shape and see how they are actually put together and their interactions. And so here is an example of a protein, topoisomerase, and you can see the three-dimensional structure of it. DNA, nucleic acid, another one. So there are four main kinds. There are carbohydrates, lipids, proteins, and nucleic acids, and we'll break each of them down in the different sections here and sort of highlight some of the main functions of them as well as what makes them up. Except for the lipids, the macromolecules or the biomolecules exist in two different forms. We have a monomer, which is the simplest, and polymer, which is repeating monomers. Mono, the prefix mono means one. I think I actually have that hanging up in my classroom. Poly means many. So if you are good with prefixes, this will help you. I will try to get you a prefix form that you can keep with you down the road, because if you are comfortable with your prefixes, your Latin roots, your some of your Greek's roots and prefixes and suffixes, then this will help you out a lot. So the movement between one unit and multiple units is facilitated by adding or removing water, depending on which way. So we're going to talk about that important part of life, the water molecule. So now that you know a little bit of water molecules and their properties, we're going to talk about how they're involved in different reactions. Dehydration synthesis. This is when we take and we build molecules together. We build it by removing two hydrogens and one oxygen from the two different molecules. So a hydrogen from one and oxygen from another, and we're able to connect it together. In the process, water is produced. So dehydration. D, think of out of water. Water is coming out of the synthesis, the formation of a new molecule. And I think I have syn. I have syn. The prefix syn is on my board. It means to build. So as we're building something, we are removing water. So that's how you can remember dehydration synthesis. Okay, so here's a little close-up of it. On the flip side, we have hydrolysis. If we are taking a bigger molecule and breaking it apart, to lyse something means to break. So when a cell lyses open, it breaks open. Water is needed. It sort of cuts it, is how I think of it. But in reality, it's going to, the bond is going to be broken, and then hydrogen will be formed on the end of one, and a hydroxide will be added to the end of the other. It reduces the complexity. We call these catabolic reactions. When we build something, it's anabolic. When we break it down, we reduce it in complexity, it's catabolic. And sort of the way I remember it is the cat scratches up the furniture and all of our furniture is destroyed because it's tore it down and tore it apart. And so these, at least my cats do that. And so it's a catabolic process because it's breaking it apart. These processes, even though we're adding water, we are needing a molecule. It releases energy, exergonic. And so energy is released, and oftentimes you need an enzyme to get the party started. It's still required in order to facilitate the reaction happening. Okay, so we're now going to take a tour of them, look at structure and function. You want to know what the building blocks are, the monomers, the different atoms or elements needed, and then what are some examples of the different polymers, the longer examples of the molecules. It's easy to get confused. Don't hesitate to ask questions. This might be a section you might want to make some flashcards for. So we're going to start with carbohydrates. Carbohydrates are the energy, the fuel. They're sugars and starches. They're made out of C, H, and O, carbon, hydrogen, oxygen, and a 1-to-1 ratio. And so depending on if you're starting at a monomer or if you're looking at larger carbohydrate molecules, if you break it down into its simplest ratio, it will be in a 1-to-1 ratio. It's used for short-term energy storage and structure. We have monomers like glucose or monosaccharides. And then we can build longer ones up like chitin and glycogen. We have polysaccharides. Different sugar monomers can have different numbers of carbon. Just keep that in mind. Okay, so here's a couple of examples of different sugars. We have pentose right here, which is part of our DNA and RNA. We have hexose, which is important for energy and structure. If you recognize that it has an ose at the end, it's a sugar, short-term energy. Monosaccharide is the name of the building block. If you see that, you know you're dealing with sugars again, the 1-to-1 ratio. Those are some of the highlights. And so here are some examples here of different monosaccharides. You don't need to memorize what a ketose looks like or an aldose, so just being able to recognize them is what's more important. There are monosaccharides. Oh, there's also disaccharides. Di prefix means two. Hexose sugars are the most famous monosaccharides. Those are those ring shapes that we're used to looking at. And glucose is probably the one that we encounter the most. Probably galactose and fructose as well. They're typically shown as carbon rings, although they are not necessarily always going to be carbon rings. You can combine two of them. Remember, if we're combining it, it's a synthesis, it's a dehydration synthesis, we get our disaccharides. Don't forget, dehydration synthesis out of water. The water is coming out. So when we take two glucose molecules, we have maltose, malt sugar. Glucose and fructose make sucrose, which is table sugar. And glucose and galactose equals lactose, milk sugar. Recognize these types. Recognize sucrose and lactose as types of disaccharides. You don't necessarily need to know the exact formula, but again, become familiar. Don't let it scare you when you're reading. Know that it's more complex than just a glucose, and that they have common properties. This is the milk sugar that is found in the milk that we drink. And in fact, a lot of people, if you know someone lactose intolerant, they have trouble breaking this lactose back down into glucose plus galactose because they don't have the enzyme to do so. Okay, and so here's an example. Most of them are in the ring shape, but not always, of the hexoses. And then you can look at the dehydration reactions if you want to a little bit more closely. Don't freak out when you see these reactions. This is not a chemistry class. We want to get the idea of it. We want to see the water leaving, but we're not going to focus on memorizing the structural details. Okay, so we have monosaccharides 1, disaccharides 2 of them have come together, and now we have when lots of different monomers have come together, we have the polysaccharide. Glucose polymers have two main functions in organisms. We have energy storage and structural support. Polysaccharides are great for storing energy in the short term, and I say short term because we're going to talk about lipids, which are a little bit more long term. In plants, amylose is the major energy storage, and glycogen in animals. And actually, know this part right here. Animals use glycogen for energy storage. We'll talk about glycogen later on when we're looking at cellular respiration. Glycogen is a molecule that you'll want to know, and also amylose being part of for plants energy storage. So these are two that you'll want to know. We might do a lab. We probably won't. But there's the labs that we can do looking at amylose. But glycogen for sure and amylose, if you don't know those terms yet, please put them on flashcards. Please highlight them in your notes. Cellulose, I know at least my freshman classes talked about it, maybe yours did too. Cellulose is the main component of plant walls. And I think of it celery. You know, celery, when you bite into that celery, it's all stringy and it's got those fibers that just don't break apart. That's due to the cellulose. So I think cellulose is celery strings. And so that structural support is what those strings kind of represents. The difference between starch and cellulose is the linkage between the glucose units. And so alpha linked and beta linked, it goes down to the chemistry. Let me see if it goes through a little bit quicker or closer. Right here, we have starch and cellulose, and all it depends on is how it's linked together. And so if you learned, I don't know if you learned in chemistry or not, the position of the carbons, we number those like 1, 2, 3, 4, 5, if you remember numbering them, but it's just how it's linked together. Don't worry about the beta or alpha linkage. And so cellulose is the structural support. It's really tough. It's a polysaccharide. It's kind of hard to break down. The problem, though, is that herbivores who rely on these celluloses in plants, they need to digest it. Animals lack the enzymes necessary to break these linkages between the cellulose molecules, and so they use different strategies. And you can think of these strategies as being both system-oriented, as in looking at the digestive system and the parts that make it up, but they also relate to evolution, because these processes that are employed have resulted by many different changes that have accumulated depending on the needs of the environment, what the adaptive advantage has been over time. And so like termites that eat wood, they have a symbiotic relationship with the protist that helps break it down. The protist lives on the termite, and and in return for the shelter, the protist helps break down the cellulose. In ruminants such as cows, I don't know if you've heard that a cow has 4 stomachs. It actually has multiple places in the stomach that help break it down. But before it even gets to that point, it is actually regurgitated. So cow eats the grass, it comes up, oops, it goes back down, it's chewed back up again, it goes back again, and then it continues on in the multiple stomachs. That's called chewing the cud. So if you ever see cows doing that, moving their jaws, they are chewing their cud. It spends most of its day doing that. So eating, regurgitating, chewing it back up again. And then you have cecophores, and these are really cool. So I don't know if you guys knew, a couple years ago we had Johnny the rabbit, or if you've seen any other rabbits, oftentimes they have 2 different color pellets of poop. And that is because they will actually, the first time food passes through, they can actually eat those little pellets that come back out a second time to get all the nutrients out of it. And so it's a way to put partially digested cellulose back into the animal. So it's kind of funny because we always joke around that the bunnies eat their poop, but it's called a cecum and are not. Oh, I forget. Actually, I have to look it up. What it's called. But I'll double check what the name of it is. But they actually do eat the pellets to get more nutrients out. Now, if food is plentiful, they might not. They might just eat fresh stuff. But if not, they can re-eat it. And you can tell the difference because the one full of nutrients that can be eaten again is a very dark color. It's healthy color. And the other one is a dried looking green color. So, cecal, not cecum, cecal pellets. And so here you can see the cecal pellets, and then this one's able to be eaten. They look like little dark, really dark colored raspberries. When they come out smoother, rounder, and it doesn't show it in as good of a light, but these are usually a lighter, like brownish green color. They look a little bit more dried. They don't eat this one, they eat the cecal part. There's other carbohydrates such as chitin. It's a modified polysaccharide. It's used in cell walls and in exoskeletons. If you've heard of chitin, it's probably with arthropod exoskeletons. It also dissolves stitches. It's one of the properties. Thank you, biotechnology. Peptidoglycan is another modified polysaccharide. It's part of the cell wall component of bacteria. And so there's other carbohydrates that you might hear about or encounter through your reading or through reading of articles. Second group here is lipids. These include fats, oils, waxes, mostly C, H, and O, carbon, hydrogen, and oxygen. It's used for long-term energy storage as well as insulation. That's another important part of it. There are no polymers. They're just big, long chains. They don't start off as simple units. They are just long chains of carbons and hydrogens. There's three main groups: triglycerides, phospholipids, and steroids, type of hormone. So triglycerides are made of one glycerol group and three fatty acids. Glycerol is this group right here. Recognize that glycerol, if you see glycerol, it's going to be going with a fat, a lipid. And fatty acids are long chains. A fatty acid is a long chain here. And it's considered not a polysaccharide because these chains are not connected together. They're connected to a glycerol that kind of hooks onto them all. Because they are being synthesized, put together, water will be released. It's dehydration. You don't need to remember the ester leakages. Whoopsie. Now, with these triglycerides, we have saturated versus unsaturated. And what that refers to are these bonds between the carbons. Saturated means that there's no double bonds between the carbons. Okay, they all have a nice stable configuration for the carbons. Unsaturated means at least one double bond. What that does is if there's a double bond, it's like a little kink in it and it influences its shape. And as we know, shape and function go together. So if it changes its shape, it's going to change its function. When we're looking here, okay, we have a saturated fat and we have an unsaturated fat. And you notice that the saturated fat can actually stay solid at room temperature. Okay, it's more packed together. But on an unsaturated fat that has a double bond, it becomes kinked. You can't pack them as tightly together. And at room temperature, you will have a more chance of a liquid form. Another group of lipids are the phospholipids. These have to do with cell membranes. We're going to be spending a lot of time on them. They are modified triglycerides. And so you take one of those fatty acids in the last one and you replace it with a phosphate group. And so you still have your glycerol group, but instead of a fatty acid over here, you're going to have a phosphate group. These come together, they form the bilayer of the cell membrane, they act as insulation, they also act as kind of a screening device, a security gate for materials going in and out, and we'll talk about that more in probably about a month. Okay. What's cool about the phospholipid bilayer is that all of these lipids with all the carbons and hydrogens, they are not polar. They do not like water. They are considered hydrophobic. They do not have free oxygen, electronegative atoms on them that are going to mix with other water molecules. They are going to stay well away from water molecules. Water molecules are not going to be able to get in between. They are hydrophobic. On the flip side, that phosphate or the phospholipid at the other side, the phosphate group, it's hydrophilic. There is room for that water to come around and sort of get in between there. It likes water. It's hydrophilic. Hydrophobic does not like water. Hydrophilic loves water molecules. And that contributes to the properties of the cell membrane. A third class of lipids are the steroids. We have a class of hormones, and we also have cholesterol. The notable structure from them are fused rings. These rings that are kind of fused together like this. This is a sign of a lipid, and it's a sign of a steroid. It has different functional groups that kind of distinguish them from one another and lead to different functions. And so here you can kind of see we have the same general shape in each of these, but we have different functional groups. And if we're looking like right here, notice that we have an oxygen double bonded, but here we have an alcohol. Okay, I want you to look at how similar these two molecules are. Okay, because these are an estrogen molecules over here, testosterone over here. So two different types of sex hormones and very similar structures, just tiny details, tiny different big difference in the functions. Proteins I call the minions of the body. They go and do everything. They are like the little go-getters. They're the most complex of the molecules. They're made up of CHO as well, but also nitrogen and a little bit of sulfur. They're used to accomplish all life functions. They're the putter togethers, taker aparts, deliverer of messages, shower of traits, etc., etc. All proteins are polymers of amino acids. So the building blocks, the monomers of a protein are amino acids, and they are joined by what's called a peptide bond. You want to know that peptide bonds are the bonds that connect these. You will want to know this, and we're going to do an activity in class in which we take different amino acids and we form the peptide bonds, so that way you can actually see how this comes together. I think, let me see if it shows it. No. So the general structure of an amino acid here is that we're going to have a carbon with a nitrogen and another carbon, and then we have a functional group. So here we go. We have a central carbon, nitrogen, the R stands for functional group, and another carbon. If you see NCC, you're dealing with an amino acid, and they're going to be coming together to form longer chains called polypeptides. Polypeptides because they are made out of peptide bonds. And since these are little ones coming together, we're synthesizing again. Since we're going to be synthesizing, we're going to do the synthesis reaction we know is dehydration, in which water is released. There's 21 known amino acids used in biological systems, and they all contain the this is a good one. The amino group, which is with the nitrogen, and the carboxyl group. Right, here's our carboxyl group, the carbon, single bond oxygen, double bond oxygen, around a central carbon, which is called the alpha carbon, and a R group. The structure of the R group varies. And this is kind of like when we were talking functional groups in the last one. Don't memorize the structure, but you need to be able to recognize by looking at it what category it would fall into, because that category that it falls into is going to dictate how its shape will look at the end, how it's going to come together with the other functional groups. And so if we're looking here, we have amino acids with electrically charged side chains. Okay, and so we have charges here. We have a positive here. We have a positive here. And so we have side chains that are considered positive. And what that means is that these have a lot of positives going on. We are going to be attracted to negative – amino acids, such as ones with these hanging out oxygens. Remember, oxygens will carry the negative charge, more the negative charge. And so we have all these free oxygens. They'll be attracted to the positive hydrogens. Remember, it's kind of like ionic bonds or what electronegativity. Think positive, negative, opposites attract. Okay, we also have amino acids with uncharged side chains. Now here we still have a nitrogen and a hydrogen, but it's not just hanging out at the end, and that's what the big difference is. Here we do have it hanging out at the end, but you also have like the other oxygens. It's not so far away as these are. Okay, we have a couple of special cases. We have cysteine. Cysteine we'll get to know. If you see a sulfur there, we have cysteine. And then we have a few other ones. Cysteine is the only one I really want you to be able to recognize without a doubt when you see sulfur, and it's because of the way the proteins form structures. We have hydrophobic side chains, in which if you look down here, we don't see anything that's going to bond with water. We have no polar molecules. Okay. Chains of amino acids have a directionality with N-terminus and C-terminus. And so the N-terminus means just the side that has the nitrogen at the end. The C-terminus is, if we're looking at the protein, the side that has the carboxyl group at the end. Okay, so that's just what that means. It's nothing new. It's just a term to describe what's at which side to look at. Protein structure will model in class. Because of the diversity of the amino acids in those R groups, they have a huge array of 3D structures that they can form. And in fact, there's such a vast array that scientists don't know them all. And they're constantly discovering new proteins and how they form together and fold back on each other in the interactions. There's a game called Fold It that you can get, an app, and it's actually university and government research agencies that are recruiting people to try to figure out how the proteins are going to fold based on the interactions and kind of try to do some of the work for them. So when we're talking about the protein, we have 4 different structures. We have the primary structure, which is just the order of the amino acids. It's the sequence. So when you learned in regular biology how to use that codon wheel and you had to find the 3 codons and you wrote down the letters to figure out which amino acid, that's your primary structure. Okay. The secondary structure is when you start seeing a couple of the interactions with hydrogen bonds. So hydrogen bonds between the atoms in the carbon and nitrogen backbone. Remember, there's the C terminus and the N terminus at the end. Okay, now think about in the middle, the carbons and the nitrogen. The way they fold on each other can either be done in a helix way, alpha helix, or the beta pleated sheet. The thing is, this is a very not misunderstood. It's a little is known about how to predict whether or not it's going to form alpha helix or the beta pleated sheet. This is kind of a fuzzy area in molecular biology, in they call the study of proteins and protein formation proteomics. Proteomics, kind of like genomics, proteomics. This is an area where there's a lot of research that still needs to be done, so we can't necessarily predict it. The tertiary structure, well, this is the part that we'll model. It's when we take that chain, it's kind of been spiraled up into an alpha or pleated into a sheet. But then what's going to happen is all these interactions between the R groups are going to happen, and it's going to create a 3-dimensional structure, a 3D shape. And so we'll have disulfide bridges forming, where 2 cysteines will meet up and form an interaction. We have hydrophobic interactions going on where the hydrophobic R groups are going to hang out with other hydrophobic R groups. We're going to have acid base. We're going to have positive negative charges coming together. And because of all the different chemical interactions happening, they make cool different shapes. And so here's an example of a hydrophobic interaction. Here's the negative. If there's a positive hanging out somewhere, it's probably going to be attracted to it. Here's a hydrogen bond formation and interaction. And this is what's called a disulfide bridge because it's two sulfurs and a bridge between the two. And then lastly, we have quaternary. So when one peptide chain, polypeptide chain, makes a three-dimensional shape, it is called tertiary. But when you have multiple polypeptides coming together with their 3D shape, you have quaternary. An example of quaternary is hemoglobin. You have multiple polypeptides that come together and form a larger unit. And in this case, this particular picture right here, what we have is transthyretin, and it has 4 different identical subunits that are coming together, so 4 different polypeptides. Question is, why do some proteins consist of more than one polypeptide chain? What do you think this allows? This is a question that I'm not going to answer necessarily right now, but to think about when you're doing your reading and looking at some of the case studies that we come across. So what do they actually do? Generally speaking, they are the go-getters. They're responsible for all life activities. Your text gives a pretty good overview in its charts. They store things. There are hormonal proteins. So we talked about lipid proteins, lipid-based steroid hormones, such as the sex hormones, estrogen and testosterone. But there's also hormonal proteins right here, like insulin. We have structural proteins for support, defensive proteins for disease, transport proteins, receptor proteins for communication, contractile motor proteins for movement, and then we have enzymatic proteins to get reactions started. Sickle cell anemia is an illustrative example of a protein. It's a relationship between protein structure and the physiology. We'll talk more about sickle cell anemia when we look at evolution. It's not the only good example, easy example to visualize this relationship between structure and function, but it's a nice one to start with. So this is hemoglobin. It carries oxygen in your red blood cells, so it is crazy important because we know that we need the oxygen. We need the oxygen for cellular respiration. Some unlucky folks, though, have a mutation that results in valine, which is hydrophobic, replacing glutamic acid, which is hydrophilic, in the beta chains of a hemoglobin. Because of that, they're going to behave differently, and it's going to have interactions that are different with the other functional groups in the chain. So in this case, normal hemoglobin, here's the beta subunit. It looks like this as compared to this. And it's all because of the hydrophilic structure here. And when it comes together for the normal hemoglobin, we have our multiple subunits. It functions. Oxygen's able to fit right here in the middle, bond right here in the middle, and it's able to travel. In this case, when it creates its hemoglobin, it's the wrong shape. Oxygen is not able to bond due to its shape, and it's not able to be carried. Sickle cell hemoglobin gets clumpy and the red blood cells change. They don't carry as much oxygen. They get stuck in the blood vessels. And those with sickle cell anemia die at a young age from the disease. That's just one of the outcomes, and it's crazy because when we look at evolution, we'll find out that there actually is you would wonder why something like that would be an allele for that sickle shape would actually be in our genome, and there's a reason for it, evolutionarily speaking. And in fact, there's a reason for a lot of things that we consider detrimental to our health. We wonder why it's even part of our human genome or an allele possibility. And a lot of it has to do with the evolutionary history. And there's a book called Survival of the Sickest, and I'll bring it up later on, that goes into a lot of these seemingly disorders that you wonder why they haven't died out, why they're still around. And the reason is because there is something positive that they have provided at some point. Now, when an enzyme or any protein becomes non-functional, it's often because of denaturation. There is a direct relationship between a protein's conformation and its function. So if you change the conformation, you change the function. When we're talking about changing the structure of a protein, we call it denaturation. Denatured proteins do not work out well, if at all. And so we unravel it, the shape changes, the function's going to change, and it won't be able to do its job. Because of their complexity, studying protein structure and functions, proteomics can be overwhelming. So that's why we use computer software. And I can show you some of the computer software, and I'll show you the folded game too, in case you would like to give it a shot. But there's just different ways. Here's a ribbon model. So you can just see the alpha. You can see the alpha helices and the beta pleated sheets. Here's a space filling model, so you can see the actual molecules. This one's more to focus on the different units that come together. By only focusing on the interacting elements of the flu virus and antibody, scientists can better understand the interactions. So depending on the needs, what you're using it for, different models might be chosen. Believe it or not, plenty of proteins can be seen with the naked eye. One of the strongest materials on Earth, if we're looking at tensile force right here, is the silk of the spider. It's very few substances, even synthetic substances made in the lab, have the tensile strength and the flexibility of this protein, the specific protein right here. Last information or last group is the nucleic acids. They are made out of SNOP, carbon, hydrogen, oxygen, nitrogen, and phosphorus. There are two different kinds, DNA and RNA, and what they differ in are their types of sugar and one of their bases. So DNA has deoxyribose, RNA has ribose. The bases are identical, except if you remember, DNA has a T for thymine, RNA, uracil. Also, the third difference here is DNA is double-stranded helix. Yay, helix. We have a helix now versus RNA, one strand. So I guess our lizard would be more of an RNA molecule rather than a DNA molecule. DNA serves two functions in all of life. It stores the information and it is heritable, so it's able to be transmitted to offspring. Okay, so the storage and the transmission. The DNA structure, we have covalently bonded nucleotides from the sugar to the phosphate with hydrogen bonds connecting them in between here. We'll get down into the structure more so towards the end of the semester. You probably heard about Watson and Crick, probably a little bit about Wilkins and Franklin. I'll bring them up again as well. Important in the discovery of DNA. A lot of controversy about these they call them lazy fools, even though they're the ones that have been attributed all the glory that goes with the discovery of the double helix model. There were actually others that were working as well. Okay, and RNA will serve many functions for life. It transmits and translates DNA info into protein. Most of the time, it's going to act in an enzymatic and regulatory function. It's either going to be working to transfer the information from DNA down to the ribosome in the form of messenger RNA and putting the tRNA that is going to be bringing in the amino acids to put them together at the ribosome, creating the proteins, or it's going to act as an enzyme. We call them ribonucleases. That's one example of RNA that is actually a enzyme, and we have quite a few of them inside of the nucleus that we'll talk about. The RNA structure is less stable because it is one-stranded, but you can still have base pairing occur. It just folds back on itself. The central dogma we'll go through next semester when we look at molecular. The central dogma is just how do we get from the stored information to functional proteins that are actually going out and doing something. And so that's how we're going to start semester 2. We won't go into details now. Make sure you can, at the end of this, identify the structures of the monomers and polymers. And when I say identify, you do not have to draw it out exactly, but you need to be able to recognize them. If you see different images, you need to know if it's a lipid or if it's a carbohydrate based on the structure. You need to be able to diagram the synthesis and hydrolysis of carbohydrates and polypeptides. Again, it doesn't have to be an exact formula, but you have to understand where the hydrogen and hydroxide groups are coming from, or where they're going to. You want to explain the biological functions of all the molecules. What's the big deal? Okay, why do we care about them? What do they do? Explain the emergence of all 4 levels of protein structure. Okay, so when it says emergence of all 4 levels, how does the protein structure change? And eventually, what does that change allow for? And finally, describe the general role of nucleic acids in living systems. What's the purpose of nucleic acids? What do they do? If you have any questions, let me know. We will probably do one more little small one on ideas for the origin of life, but this will be the bulk of your test material right here. So on the last one, know water. Spend some time on chapter 3. Know why carbon is cool and what sets it apart from other atoms and why it's important in living systems, and get an idea, start getting a grasp on the different macromolecules. We'll spend a little bit of time on enzymes later on. We'll spend a lot of time on proteins and nucleic acids. Not as much time on carbohydrates, and we'll look at lipids when we look at cellular membranes. Okay, hope you guys have a good day.